Gaseous exchange in plants. Gaseous exchange in plants is the process by which they take in carbon dioxide CO2 from the atmosphere and release oxygen O2 into it through the process of photosynthesis. Photosynthesis occurs in specialized plant cells called chloroplasts, which contain chlorophyll pigment that absorbs light energy. The overall process of gaseous exchange in plants involves the following steps. 1. Stomatal opening. During the daytime, in the presence of light, the stomata, specialized pores on the leaves and stems of the plant, open to facilitate gas exchange. 2. CO2 uptake. As the stomata open, CO2 diffuses from the atmosphere into the plant through the stomatal pores and into the leaf cells. 3. Photosynthesis. Within the leaf cells, the absorbed CO2 is used in the process of photosynthesis. Light energy is absorbed by chlorophyll pigment and is used to convert CO2 and water H2O into glucose, C6H12O6, and O2. 4. O2 release. As a result of photosynthesis, O2 is released into the atmosphere through the stomatal pores. Overall, this process of gaseous exchange in plants plays a crucial role in maintaining the balance of gases in the atmosphere by taking in CO2 and releasing O2, which supports the respiration and survival of other organisms. Importance of gaseous exchange in plants. Gaseous exchange, particularly the process of photosynthesis, is critically important for plants for several reasons. 1. Energy production. Photosynthesis is the primary process by which plants produce food in the form of glucose. Glucose is used as an energy source for various cellular activities, such as growth, maintenance, and reproduction. 2. Oxygen production. The process of photosynthesis enables plants to release oxygen into the atmosphere. Oxygen is an essential component for the respiration of animals and other organisms, which in turn helps to maintain the balance of the atmospheric gases. 3. Carbon dioxide absorption. Plants take up carbon dioxide from the atmosphere during the process of gaseous exchange, which helps to regulate the levels of atmospheric CO2. This, in turn, helps to mitigate the effects of climate change. 4. Water conservation. During the process of gaseous exchange, plants also lose water through the stomatal pores. However, they are able to regulate the rate of water loss to preserve water and maintain cellular hydration. Overall, the process of gaseous exchange in plants is essential for their survival and plays an important role in regulating the Earth's atmospheric gases and supporting the ecosystems. Gaseous exchange in the leaves. Gaseous exchange in the leaves of plants primarily occurs through the stomata, small openings or pores on the surface of leaves and stems. The process of gaseous exchange in leaves involves the following steps. 1. Stomatal opening. During the day, in the presence of light, the stomata open to facilitate gas exchange. This allows for the diffusion of gases between the atmosphere and the leaf's internal tissues. 2. Carbon dioxide uptake. As the stomata open, carbon dioxide CO2 diffuses into the leaf from the atmosphere and into the leaf's mesophyll cells. 3. Photosynthesis. Within the mesophyll cells, the absorbed CO2 is used in the process of photosynthesis, which is powered by light energy. Light energy is absorbed by chlorophyll and is used to convert CO2 and water into glucose and oxygen. 4. Oxygen release. As a result of photosynthesis, oxygen O2 is released into the atmosphere through the stomatal pores. 5. Water vapor release. Water vapor is released from the leaf through the same stomatal openings in a process called transpiration. This process helps to regulate the water balance of the plant and maintain cellular hydration. Overall, the process of gaseous exchange in leaves plays a vital role in maintaining the balance of atmospheric gases and in supporting the growth and survival of plants. Fig. 5.6. Stomata. Mechanism of closing and opening of stomata. Stomata are small pores found primarily on the surface of leaves and stems in plants. They are responsible for regulating gas exchange between the plant and the atmosphere. The opening and closing of stomata are controlled by specialized cells known as guard cells, which are found on either side of the stomatal pore. The mechanism of opening and closing of stomata involves the following steps. 1. Opening of stomata. When there is ample light and the plant has enough water, the guard cells actively take up potassium ions K+, from the surrounding cells. This causes the concentration of K+ ions to increase within the guard cells, which decreases the water potential of the guard cells. As a result, Water enters the guard cells by osmosis and the cells become turgid, causing them to bow out and the stomatal pore to open. Figure 5.7. B. Stomata open. 2. Closing of stomata. When conditions become unfavorable, such as during drought or at night, the concentration of K+ ions in the guard cells decreases, causing water to move out of the guard cells and they become flaccid. Thus, the guard cells become less turgid, and the pore between them closes. This is a result of the loss of water from the guard cells. Figure 5.7. A stomata closed. Thus, the opening and closing of stomata is a dynamic process that is regulated by various stimuli such as light, CO2 concentration, temperature and humidity, among others, and ultimately helps the plant to regulate its water balance and gas exchange according to its environmental signals. Gaseous exchange in stems. While gaseous exchange primarily occurs in the leaves of plants, it can also occur in the stems, particularly in those stems that are young and actively growing. The process of gaseous exchange in stems involves the following steps. 1. Diffusion. The exchange of gases, primarily carbon dioxide and oxygen, occurs through the stem surface by diffusion. 2. Intercellular spaces. Within the stem, there are intercellular spaces that allow gases to move from one cell to another. 3. Lenticels. In some stems, a specialized structure known as lenticels can also facilitate gaseous exchange. Lenticels are small, raised areas on the stem surface that allow for gas exchange between the internal tissues of the stem and the atmosphere. Fig. 5.8. Lenticels. 4. Secondary growth. In older woody stems, diffusion alone is not sufficient for gaseous exchange. Instead, secondary growth contributes to the formation of porous regions within the stem, such as growth rings and wood fibers, which allow for gaseous exchange. Overall, gaseous exchange in stems plays a smaller role compared to that in leaves, but it is still essential for maintaining the overall metabolism of the plant, especially in young, growing stems. Gaseous exchange in the roots. While roots mainly respire anaerobically, meaning without oxygen, some gaseous exchange can still occur in the roots, particularly in the outermost layer of cells known as the root hairs. The process of gaseous exchange in the roots involves the following steps. 1. Oxygen uptake. Oxygen diffuses through the soil and into the root system, mainly through the root hairs. 2. Carbon dioxide released. Carbon dioxide produced from the root cell's metabolism is released back into the soil through the same process of diffusion. 3. Aerobic respiration. Oxygen is used for aerobic respiration in the inner root cells that have more mitochondria as compared to the cells in outer layers. The main role of gaseous exchange in the roots is to ensure that the roots have enough oxygen for aerobic respiration, which is vital for providing the energy needed for root growth and development. Additionally, the exchange of gases in the root system can also be vital for maintaining proper soil aeration and microbial activity, allowing for optimal nutrient uptake by the roots. Pneumatophores. Pneumatophores are specialized roots that grow above the water surface in some species of trees and plants to allow gaseous exchange between the atmosphere and submerged roots. They are commonly found in mangrove trees but can also be seen in some other types of plants that grow in swampy or marshy areas. The main purpose of pneumatophores is to allow roots to obtain oxygen for respiration, which is essential for growth and metabolism. The underwater roots of these plants are often surrounded by stagnant or low oxygen water, which can limit their ability to respire aerobically. Pneumatophores are typically small, vertical structures that protrude above the water level, ranging from a few centimeters to over 30 centimeters in height. They often have numerous lenticels, which are small openings on the stem or root surface that allow for gas exchange between the atmosphere and the internal root tissues. Overall, pneumatophores are an adaptation that allows certain species of plants to thrive in wetland environments by facilitating gaseous exchange for underwater roots. Fig. 5.9. Pneumatophores. Revision exercise 5.3. 1. What is the importance of gaseous exchange in plants? Gaseous exchange is essential for plants to maintain their metabolism by obtaining the gases they need for cellular respiration and photosynthesis. In respiration, plants take in oxygen, and they release carbon dioxide as a waste product. This means that plants need to have an adequate supply of oxygen to carry out aerobic respiration for energy production. Additionally, in photosynthesis, plants absorb carbon dioxide and release oxygen as a byproduct, which is needed for many other organisms' survival. Therefore, Gaseous exchange in plants is crucial not only for the plant's survival but also for the entire ecosystem to maintain an adequate supply of oxygen in the air. 2. Where do gaseous exchange in stems take place? Gaseous exchange in stems primarily takes place in the small, microscopic pores called stomata, which are located on the surface of stems and leaves. Stomata play an important role in gaseous exchange by allowing the diffusion of gases between the plant and the environment. When stomata are open, carbon dioxide can enter the plant for use in photosynthesis, and oxygen can exit the plant as a product of photosynthesis and respiration. Stems also have intercellular spaces within the tissue, which allow for the diffusion of gases between cells. Overall, the gaseous exchange that occurs in the stems is important for ensuring that plant cells have the oxygen and carbon dioxide necessary for respiration and photosynthesis, respectively, as well as for regulating water loss through transpiration. 3. What are pneumatophores? Pneumatophores are specialized roots that grow above the water surface in some species of trees and plants to allow gaseous exchange between the atmosphere and submerged roots. They are commonly found in mangrove trees but can also be seen in some other types of plants that grow in swampy or marshy areas. The main purpose of pneumatophores is to allow roots to obtain oxygen for respiration, which is essential for growth and metabolism. The underwater roots of these plants are often surrounded by stagnant or low oxygen water, which can limit their ability to respire aerobically. Pneumatophores are typically small, vertical structures that protrude above the water level, ranging from a few centimeters to over 30 centimeters in height. They often have numerous lenticels, which are small openings on the stem or root surface that allow for gas exchange between the atmosphere and the internal root tissues. Overall, pneumatophores are an adaptation that allows certain species of plants to thrive in wetland environments by facilitating gaseous exchange for underwater roots. 4. Describe the photosynthetic theory that explains the mechanism of stomatal opening and closure. The photosynthetic theory of stomatal opening and closure proposes that the opening and closing mechanism of stomata is regulated by changes in the intercellular concentration of carbon dioxide and the rate of photosynthesis in the plant. During the day, when there is sufficient sunlight, plants perform photosynthesis, which requires carbon dioxide and produces oxygen. When the intercellular concentration of carbon dioxide decreases due to the uptake from photosynthesis, it triggers the stomata to open, allowing more carbon dioxide to enter the plant for photosynthesis. As the intercellular concentration of carbon dioxide increases, the rate of photosynthesis decreases, and the stomata close to conserve water and prevent excess transpiration. Additionally, under certain conditions such as water stress, the plant releases abscisic acid ABA, a hormone that signals the stomata to close, reducing water loss. Overall, the photosynthetic theory proposes that the opening and closing of stomata are controlled by a balance between the plant's need for carbon dioxide and water conservation. The mechanism ensures that the plant can carry out photosynthesis efficiently while minimizing water loss through transpiration.